So the questions that I ask myself, what kind of message I can deliver to this group? Instead of, planning is really coordination is going to be really essential. So this is the plan sort of I have outlined. Mountain pine beetle tree colonization it goes through several stages, from dispersal to all the way to the reproduction and offspring emergence. So what I'm going to do, I'm going to look at those essential, some of the chemically, behaviorally important chemicals at each stage. At the end, I will put together, and Violet and I have 2 case studies. One of them I'm going to present it here. Using those chemical information, try to explain the range expansion of mountain pine beetle, as well as the role of chemical ecology to understand the survival of lodgepole pine trees during mountain pine beetle attack. What we have, we know that beetles emerge, the offspring beetles emerge there from natal hosts, and everything starts from that point. After the second step is really these beetles, after emergence, their goal is to locate most suitable host. And there's a lot of complex factors involved in this process, I will say decision-making process. It's really not clear. But I would like to tease them out little by little. And in this case, this stage is really important, especially understanding the role of aggregation pheromones produced by beetles. And the third is mass aggregation. This is the stage is really characterized by the overcoming tree defenses, basically whether insects will lose the battle or the trees. Depending on the outcome, the fourth last stage is the host establishment and reproduction, I call it. This is the case is really important. In this case, the tree lost the battle and then insects are successful, but this is not the case all the time. Now, this is the question. This is the forest, really beautiful green conifer forest, but we have this kind of picture on the landscape now. All these kind of factors, what I'm going to do, I'm trying to explain these landscape level processes based on the stand level and tree level interactions between insects and trees. The first stage is, before going forward, I want to talk about a little bit these plant chemical compounds. Plants have a lot of chemicals. Some of them we call primary metabolites. These are primarily important for growth, reproduction, and development, and including photosynthesis, respiration, and protein synthesis. But the other second group of compounds we call secondary metabolites, they don't have much function in the basic plant function. What seems to be important primarily in evolutionary or ecological interaction with other plant species, and one thing that I highlighted here is that really their adaptive significance in protecting trees against insects and the pathogens. There are a lot of chemicals they have, phenolics, terpenoids, and alkaloids. Alkaloids is really, this is a lot of chemicals, fun groups or chemicals are here, we use daily actually. And then phenolics and terpenoids. Oh, when I say daily, I didn't really mean the cocaine here. Some other compounds, that caffeine, for example. Just, I want to make sure. It's a lot of chemicals they do, and the terpenoids is really the most diverse of those three. But when we look at the plant-insect interactions, we see these opposing roles of these plant chemicals. One cases, they are attractant for insects. In other cases, they can be repellent. In some cases, they can be highly beneficial to beetles. In other cases, they can be highly toxic. Pretty much, the system is really driven by some individual compounds as well as their concentrations inside the tissues. I'm going to tease them out little by little, and then you will get the pictures. This is the table that I put together to understand what are the roles of pine chemicals in mountain pine beetle activity. These are mainly terpenoids compounds, basically, monoterpenes from alpha-pinene to all the way through carine. And we have some phenylpropene here for alisanol. Others are, we know, the fatty acids, basically. What do? These chemicals play different function at the different stages of insects. They can, like alpha-pinene, they might precursor the female aggregation pheromone. In other cases, they can be anti-fittant, especially for alpha-pinene. If insects are introduced at the higher concentrations, they can be anti-fittant. But in general, we call pheromone synergists, that these are the compounds such as terpinolene and myrcene help beetle to colonize trees. And these are the chemicals come from the trees itself. But others are more toxic. I will say limonene is one of the most toxic compounds based on the most recent research. And we have also compounds like tricarine and some other fatty acids can be highly toxic to beetles. Now, what I'm going to do, I'm going to put these compounds, look at this, you don't need to memorize anything on this table, put them together little by little, put everything in the different attack sequence of the beetles. What we have, this is the emergence and dispersal. This is the first stage. In this case, this is important. Natal host quality really matters. In terms of phloem thickness and the content, nutritional quality, what kind of impact they can have. They can introduce, they can have an effect on offspring production, numbers per mated pair, as well as the offspring quality, size and energy reserves that the beetles coming out from the tree, it affect their decision-making process. And some species overall are favored by mountain pine beetle, others, and apparently white pine and the lodgepole pine seems to be really a suitable host for beetle, even though beetle evolved with other host species. Still in this case, after they emerge right now, they are in the air. What are the factors that affect host colonization after emergence? I tried to put them together, a few items here. Species composition is really important. Whatever the forest, we have a mixed species. I'm not talking about just different pine species. I'm talking about different conifers and hardwood, mixed wood actually, will have a different impact on the insect. Host density and connectivity, we can see it here. If the forest stand up composed of very large trees and dense trees, they tend to be more susceptible to insect. We have age classes. We have a forest with a single age cohort or a mosaic age cohort. They have a different impacts on the beetle attraction. This is the point that I would like to get back to it. The trees release a lot of chemicals. Some of them are attractive, some of them repellent. And these are the usually tend to provide long-range cues. Here, this is the picture you might see. This is not ozone, it's not air pollution or anything. These are the chemicals that released by the trees under certain conditions. We don't see it. Unfortunately, in Western Canada, we don't have that kind of facilities that we can measure. We call a tower that's set up inside the forest that we can measure how much chemicals are coming out from the forest, because forests can release some of the nastiest compounds such as methane and others. These are the mainly terpenoid compounds. Here, this study to look at it, how much some forest types, these are conifers, they release a lot of isoprene, monoterpenes, and sesquiterpenes a lot. They can stay, some of them lifetime is really short, but they continuously produce the same compounds over and over again. Do they have consequences for insect attraction? Absolutely. Do we know anything about mountain pine beetle? No, at this stage. We need to tease out, this makes sense, why certain forest stands are more attacked by mountain pine beetle versus others. The second stage is host location. Right now, insects fly, they emerge from their natal host, and they are closing to their target tree. We call primary attraction is really important in this case. Primary attraction is primarily driven by the plant volatiles, right? The all volatiles, majority of volatiles that trees release, they come from the needles. Some of these compounds are attractive here, beta-phallandrin, terpene, and myrcene, while others can be repellent. Again, these are really short distance occurring around the tree. Here, this is the diagram shows that what kind of chemicals that at the stand level the trees can produce. Here is the conifer, they produce a lot of terpenoids here. Other hardwood trees, for example, they release green leaf volatiles. They tend to be inhibiting beetle attraction. Besides that, actually, we also done a lot of work on this. The soil microorganisms release a lot of terpenoids compounds too. The role of these compounds currently, we really don't know much in beetle attraction, but nevertheless, they all make up the blob of the chemicals that I showed before. It's part of that whole unit. Now, the beetles landed on the tree. What they gonna do? This we call secondary attraction. This primarily this whole system driven by female beetles. They attract it. They locate the most suitable tree. They enter the tree and they start understanding whether that tree is suitable or not. Based on that, they produce pheromone or not. There is a positive dependence between the content of the phloem they're eating versus the pheromone that's produced by female beetles. In this case, again, some chemicals can synergize, means improve the beetle attraction. Other cases, they inhibit. Terpinolene and myrcene can improve beetle attraction. For alisanol, for example, they inhibit the beetle attraction to its own pheromone. And second stage continues. Here is really important, this process. The phloem, where the insects feed, contains a lot of toxic chemicals, such as limonene, tricarin, some diterpene resin acids, and some fatty acids. They are really nasty compounds. Nevertheless, they can affect. At this stage, in my understanding, that beetles face very heavy mortality at this stage. Most of the time, actually, these attacks fail. What are the factors that can influence this stage? First of all, I put it over the surrounding beetle densities. This is really important. Number and then distance of infested trees. Here's the defensive tree defensive capacity, especially constitutive defenses we call first line of defense. We also second line of defense, rapid and localized induced defenses. These are the first time that when the beetle attack the trees, tree try to contain the attacks, and later on, especially the rapid speed of the attacks are really, really important. And the trees with such defenses are winners. Trees with the compromised defenses are usually fails. Fungal also associates with the beetles, that beetles get a lot of fungi. They can compromise those tree defenses. Here's a very good example. Here's a very successful attack and failed attack. Everything has happened at this stage, really tree level. What is the content? What chemicals present? What concentrations present in the phloem matters. The next stage is, if this attacks the stages pass through, the third stage is host aggregation. This is here all about host chemistry, really. It's usually concentration dependent. The beetles producing pheromone, trees release a lot of chemicals, everything is coming together, and if it's successful, beetles mate, and females start excavating over position garrels, lay eggs, and the beetles emerge. What are the important chemicals? In my mind, in these stages, we have a toxic chemicals, limonene and others, for alisanol and alpha-linoleic acid. It seems a very interesting fatty acid that absolutely toxic. There is nothing can survive any low concentrations. What are the factors that affect the beetle host aggregation? Surrounding beetle densities is really important. Some of the studies funded by FRI, we call trap tree studies, the colonization, the probability of trees colonized by mountain pine beetle is really response to how many trees around that the colonized trees. They drive the density, beetle density drive everything here. And the third tier of defense I want to call is delayed induced defenses. They are still important dealing with the beetle or killing the beetles. Here, amount of resources to support defense is important. If the tree defenses are compromised, such as drought, we talk about it, or fire, or other insect disease, or insect attack, or disease attacks, they can compromise tree defenses. If that is the case, tree usually lose the battle, everything is over. Here's a very good example, successful colonization, a lot of pitch tubes. In other cases, very few pitch tubes still present, but tree won this battle. The last case is about host establishment and reproduction, and there is nothing. The tree is that this, what everything I presented here usually happens within few weeks. After that, if the game is over, the tree insects start settling in, and then you don't see much further colonizations. Right now, you see the larval development and others. In this case, again, the colonization of host tissues with fungi seems to be really, really important in this case. Again, here's a successful colonization and here's unsuccessful colonization. Again, in even the most resistant trees, beetles can still attack, they can still tunnel. One thing is missing, they cannot lay eggs. They fail. We've seen a lot of examples of this kind of trees right now in Grand Prairie, for example. What I'm going to do in these short presentations, I'm going to put everything together. Can we use such information related to mountain pine beetle lodgepole pine interactions? Further elaborate the most recent host range expansion of mountain pine beetle into jack pine. Yes, I look at the same components. Constitutive defenses, I look at it. Rapid induced defenses, I look at delayed induced defenses, and all others. What we found here that jack pine seems to be really suitable. They don't have much constitutive response. They don't have a much rapid and localized induced response. They don't have much induced responses. And plus, they have the good chemicals that insects benefit from them. The fourth one is I look at it, based on chemical similarity between jack pine and lodgepole pine, apparently allow the beetles really produces pheromones, mate, reproduce, complete its development, and disperse from the from jack pine. Exactly the same thing happening. We conducted studies, look at the suitability of chemicals from jack pine to the symbionts. We have the same pattern. Symbionts apparently doing really well. And invasion of hybrid forests, I think this is popping up more and more. This process is really, really important in the beetle. I'm going to ask Violet right now to cover the remaining ones.
Okay. Thank you, Nadir. Today I'll quickly present a case study using resin-based anatomical defenses to explain the survival of larval pine trees in post-mountain pine beetle stands. We all know after mountain pine beetle outbreak, lots of larval pine trees died, but there are still few trees alive in post-mountain pine beetle stands, and that's what we call as residual trees in this project. The stand conditions change after mountain pine beetle outbreak. However, it is still unknown if those changes could affect residual trees. And people also found the larval pine regeneration is very low in post-mountain pine beetle stands in Alberta, and that makes our residual trees more important, because they could be the only future seed bank for our larval pine forests in western Alberta. During tree defense, resin is very important. However, the measurement of resin flow cannot be repeated for a certain historical time point. Therefore, we set our mind to the xylem resin ducts, and because they are the anatomical structures of producing and storing resin. And they could be a strong indicator to produce tree survival during beetle outbreak. On the other hand, tree growth would affect tree defense as well. Therefore, we asked 3 research questions to hopefully answer the relationship among growth, resin duct-based anatomical defense, and tree survival before, during, and after beetle outbreak. In total, we selected 31 sites in western Alberta, and my targeted trees are mature residual larval pine trees. We then separated those trees into 3 categories based on their health conditions, as healthy, declining, and survived. Among them, declining trees are trees showing bark lesions, sparse crowns, or yellowish needles due to other insects or pathogens attack, but not by mountain pine beetle. And survived trees are trees showing earlier mountain pine beetle attack symptoms, but they appearing vigorous. So in total, we collected 213 increment cores from residual trees and 140 wedges from mountain pine beetle-killed trees. To understand why residual trees were able to survive from beetle outbreak, we compared the growth rate and resin duct characteristics before outbreak between residual and mountain pine beetle-killed trees. On this graph, the y-axis shows the annual radial growth rate, and the x-axis shows the first 10 years right before beetle outbreak. And the blue line represents mountain pine beetle-killed trees, and the gray breaking line represents residual trees. And we found there was no differences on radial growth rate before outbreak between mountain pine beetle-killed and residual trees. Among resin duct characteristics, residual trees have consistently larger resin duct than beetle-killed trees, and that indicates the individual resin duct area may be a better predictor of tree survival than tree growth rate and the other resin duct characteristics during outbreak. However, the resin duct size can be affected by environmental factors. So to test the influence of changed stand conditions after beetle outbreak, we compared growth and resin duct characteristics on residual trees between before and after outbreak. This graph shows a percentage change in mean radial growth rate compared to before mountain pine beetle outbreak. And the red line on 0 represents no changes after beetle outbreak. And we found the survived trees have significantly decreased growth rate right after beetle outbreak in the first 5 years. Meanwhile, we found individual resin duct area is the only resin duct characteristics showing no differences after outbreak. Therefore, the environmental change in stand did not affect individual resin duct area on residual trees, and that suggests the individual resident area could be mostly be under genetic control. So based on those results, we're now wondering if we can use individual resident area to explain the different health conditions of residual trees. So we tested the latest 5, 10, and 20 year, regardless of the time of mountain pine, the outbreak time. And we found the declining trees, which are the trees showing symptoms or signs due to insects or pathogen attack, but not by mountain pine beetle. Those trees have smaller individual resin ducts than healthy trees consistently, and also smaller than survived trees in most of the time. So as conclusion, we suggest that larger xylem individual resin duct could enhance tree resistance, and it could also be a potential predictor of tree survival, not only during beetle outbreak, but also during endemic phase of broad bioagents. We also expect the future pine forests in Western Alberta following outbreak, they could show resistance to a wide range of biotic disturbances. Thank you very much.